So 2 quick announcements before we get started. Hopefully I'm going to make through the lecture today with my voice completely dying. First one is on our right, the biology department seminar this week, tomorrow at noon, will be Dr. Naomi Ward, who will be talking about membranes and microbiomes, adventures in cell biology and ecology. So really interesting studies that her lab has been doing on looking at microbe interactions with each other and also with the human gut, which is, I think, mostly what she's going to be talking about there. Then seem to be a few fewer people here. Probably a good thing that they're hanging out with their Valentines. These are the Valentines that I would otherwise be hanging out with myself. So with that, lecture 14 on the 14th of February seems to be sort of an appropriate time here. So we're going to finish up talking about messenger RNA processing. First, a little bit on splicing, but then concentrate on polyadenylation, what happens at the 3' end of your messenger RNA, and then RNA export, how that processed RNA actually gets outside of the nucleus, and then what goes on with what you need to have to undergo translation. So it's the ribosomal RNA. That ribosomal RNA is also transcribed in the nucleus, eventually needs to get out in the cytoplasm in order to undergo translation. But again, to review a little bit of splicing, there's been a huge amount of progress literally in the last few years on understanding the structure of the spliceosome. And when I say the structure of the spliceosome, this is going to be all of the small nuclear RNAs, all the proteins that are associated with them, and your primary transcript or your pre-messenger RNA. So this is one from almost exactly a year ago, publication which came out in Nature. And the main sort of take-home message here is that we've got all of these base pairing interactions between the different RNAs. So this RNA up the top here, in yellowish, I guess, what the right term for the color is U6. Down at the bottom, in green, is U2. And then we have U5 over here. In red is the pre-messenger RNA, exons and introns. And importantly here, we now know exactly what the structure looks like when you've got this 5' end phosphate binding to the 2' OH here in the branching point of the lariat. So lots and lots of base pair interactions, and U2 here, base pairing right where we have this junction taking place. Of course, we always draw these things linearly, but they're not. They're all wrapped around each other in terms of these helices. And in order to get any of the changes, of course, you need the activity of the RNA helicases, which are mostly proteins that are part of this complex. So if we now look at the proteins that are part of this complex, it gets even more ridiculously complicated. But again, we know now at a basically atomic level where all of these components are of the spliceosome. And here, actually, to make things a little clearer, all of the proteins are in gray. And hopefully it's pretty obvious that there are a whole bunch of proteins in here. A lot of the mass of the ribosome really is a lot of these proteins, some of which are RNA helicases, but others are sort of holding each of the individual RNAs in the right places. So here we have the U5 small nuclear RNA in blue, U6 in red, really very spread out here, U4 in yellow, and then U2 here in green. This is before the loop formation takes place. You'll see it's really spread out. And in the black, which actually is not in the structure, here's our pre-messenger RNA. Here's the 3' exon. Here's the 5' exon. These guys need to get put together. How does that happen? There's this huge conformational change that happens. So going basically from here to here. It's all the same proteins, all the same RNAs, but now where this branch and the 5' exon are have been folded in together. And so now this is where you're going to get the attack of that phosphate on the 2'-OH to get the linkage to form, and then processing taking place. And again, this is all about this U6 small nuclear RNA, which has the catalytic site through the RNA binding to the metals that we talked again about last time. This was August of last year that this particular paper came out. So we're beginning to get an idea of the atomic and molecular nature of exactly how the spliceosome works. This is great, except this is the normal spliceosome. And in terms of thinking about alternative splicing, how does alternative splicing start to influence this spliceosome? And that, stay posted. The next couple of years, hopefully, we'll have some more data on some of those things. So we zoom out a lot because, again, a lot of this we only know in blobology. So we don't have the high-resolution structures for a lot of these things. Last time we talked a little bit about alternative splicing. Between 60 and 90% of the genes that we will transcribe into RNA are alternatively spliced in some way. And we're slowly learning about how some of that happens. Last time we talked about SR proteins, here confusingly labeled as RS proteins. Again, it's just arginine and serine, so it doesn't really matter whether you have it as serine, arginine, or arginine and serine. These are proteins that are very rich in both of these individual amino acids. And they bind to exons. And when they bind to exons, they will stimulate binding of U2AF, which allows U2 binding. They will also stimulate binding of U1. And you remember U1 and U2 are the ones that are determining the 5' end and the 3' end of the intron, which is going to be used. And if you have that binding, then you'll have U4, U5, U6 come in, and all of this splicing will take place. These are also known as exon enhancers. If you see these little green vertical lines here, these represent specific sequences in exons that those SR, RS proteins are binding to. So these will be an exon enhancer sequence, which means that exon is going to be used. We'll talk about enhancers and silencers for transcription later on. Silencer, of course, is the exact opposite of an enhancer. This is now a sequence in pink, which is going to bind to a different set of proteins. These are the ones that have lots of arginines but glycines and not serines. What's the big difference there between glycines and serines? OH. What happens to OHs? They get phosphorylated. They're also going to be interacting in very different ways than glycine, which basically has practically no side chain whatsoever. So if you have binding by these silencer binding proteins, and silencer, again, is that sequence in the RNA, then you're not going to have interactions with U2AF. You're not going to bind to U2. You're not going to bind to U1. So the splicing machinery is just going to go and find the next exon. And so this exon here will be skipped. This is one really pretty common mechanism. And we talked about this before. Most exons get included. A few of them can get exchanged one way or another. That's only one of the known possibilities. And this is just an overview of many of these different possibilities. And just shown up at the top is that skipping arrangement. So on the top, you skip this exon in blue, and in the bottom you include that. And that's probably due to the presence of these SR proteins and or RG proteins in that particular cell at that particular time. So you'll be making a differently spliced RNA. You can also have different splice sites that are used. That can be either at the 3' end of your intron or at the 5' end. It's actually not shown here. But you would be splicing to a different position and just elongating one of these exons. There are some cases where introns are actually maintained completely. They're not spliced out at all in a few messenger RNAs. And then some even more complicated ones where it appears, that we have either one intron which is being included and a different one. So you can either have one intron, or say one exon, or a different exon which is being used. This is this mutually exclusive process right here, or depending on where you actually start transcribing, if you're to start transcribing right here instead of over here, you're never going to have the opportunity to include this particular exon. And so that will give you a different final product over here. And then, of course, you can also have tailing that takes place in a different position. And that's going to be dependent on the splicing which takes place. And remember, all of this is happening co-transcriptionally. So the RNA polymerase is making these RNAs. And then the rest of the machinery is deciding which of these exons to use and which not. So why should we care about all of these things? Turns out that probably 10% of genetic diseases in humans are due to differential splicing that takes place. And one of the classic examples of this is beta thalassemia. One of my master's students actually had beta thalassemia, and she said she hated going to the doctor because they said, oh, cool, you've got this really interesting change. It's like, no, just take care of me. Don't bother with all of these cool genetic changes that I have. So what happens in beta thalassemia, and this is just one of the kinds of beta thalassemia, is that you have changes that can take place, and very often just single nucleotide changes, and usually a single nucleotide change is not going to make a big difference in terms of whatever protein is that gets made, unless it's involved in splicing, because then you're going to be changing a whole bunch of the rest of the protein. So normal beta globin has 3 exons. These 2 introns get cut out. You end up with all 3 of these exons. Some cases of beta thalassemia, you have a single nucleotide change, and it'll be right here at the beginning of this exon or the end of the intron. Both of those have been seen, and this exon is never used. So now we have a beta globin, which is missing part of the protein. In some cases, you can have a single nucleotide change. So instead of splicing to the 5' end of this exon, you end up splicing into the middle of the intron. And so you end up with some extra sequence here. And then in some cases, you'll end up with yet another little piece, which is added here, and yet another intron, which is being added. So single nucleotide changes, and again, we may get to this translation today. We may get to it on Friday. But single nucleotide changes usually don't make a big difference in terms of your actual final protein sequence. However, if it changes splicing, that's going to make a big difference. Yes, in the back there. Yes, so cryptic splice site, as it's defined here, that's going to be where you could have splicing with just a single nucleotide change. And so normally you're not going to have splicing there, but the single nucleotide change allows the splicing machinery. And of course, that's going to be probably, if we look at it in this case, it's going to be the splice site in the middle. So that's going to be at the 3' end of the intron. You'll have a single nucleotide change there. U2AF and U2 are going to associate at that position, which is then incorrect as far as the wild type form of the gene. Yeah?
So would that be a mutation in the proteins that bind, or a mutation somewhere else, in the U1 and U2?
So the question here, I think, has to do with, what is the actual, where is that mutation? That mutation is going to be in the DNA, which codes for your primary RNA transcript, and then that base change. If you have mutations in the U2 snRNA, you're probably not even going to be here. It's so detrimental. If you think about the number of alternative splicings that you're going to have, that's highly likely to be an embryonic lethal kind of mutation. So yeah, so these are going to be in individual genes where you find them. And so again, this was a master's student working in my lab, so clearly it wasn't a lethal kind of event which took place here. Okay. More questions on this splicing stuff. We'll talk about regulated splicing. We talk about regulation later on in the course, but that's after the midterm, so who cares? So start out with our first clicker question for today. I know it's Valentine's Day. I shouldn't ask clicker questions, should I? Let's close. The presence of which of the following factors in a specific cell is thought to determine whether an exon is used or not in alternative splicing? BBP, SR proteins, 5' splice donor, 3' splice donor, U4, U5, U6. Aiming for 100% correct. Let's get there. Ten, 5, getting close to 100% correct here. Oops, let's show the results before I actually choose the correct answer. That might help. So yes, it is the SR proteins. Yay, I guess I've explained it enough, or you all guys already knew it already. So that being said, branch binding protein is important for what? Determining where that branch is going to take place. So that's also going to be interacting with U2, SR proteins, determining what it is. And then 5' splice donor. Sometimes, I just wanted to explain this. I hadn't really talked about this too much already. So the splice donor, that's going to be at the end of the intron. So it's the 5' end of the intron. 3' splice acceptor, it's going to be the nucleotide at the 3' end of your intron. And then what do U4, U5, and U6 do? They come in after you have U1 and U2 binding, all kinds of fun rearrangements and massive numbers of proteins and crazy structural changes, as we looked at right at the beginning of today's lecture. And then U6 is what's going to do your final job of taking care of the process. So one question that came up, again, Darwin Day was Monday, but we'll continue to talk about that evolution through the rest of the course, and really fundamental for all of biology. Where the heck did introns come from? And why are they here? Probably so we can get huge amounts of diversity. Yeah? Pardon? Oh, the pole window, sorry. Hide that now. Sorry about that. So you can see something? Yeah, being able to see, yeah, who knows? Go figure. Yeah, so one of the questions arose is where the heck did introns come from in the first place? And how did they evolve into what we have now in terms of all of this splicing machinery? And this goes back, oh, I know, to the late dark ages when I was looking at graduate schools. So turns out that there were a couple of labs, particularly being led by Tom Cech at the University of Colorado, where they were looking at RNAs that they had purified, and they were looking for the enzymes that were important for doing the splicing, because that had been shown by Roberts and Sharp a few years earlier. So what's going on? What are the enzymes that are important for doing this splicing activity? So Tom Cech had people in his labs purifying the proteins that they thought were important for splicing and purifying the RNAs that they were going to use for doing the splicing. And then his students started coming to him and saying, Dr. Cech, we have this RNA and we purified away all the proteins, but it's still splicing. What? No, go back, do it again. Do it again, do it again. And I actually interviewed with Tom Cech, again, thinking about doing PhD work in his lab. And he told me this story about how he did not believe any of his students or his postdocs for about a year. And then eventually figured out that what was going on is the RNA itself actually had this splicing activity. And Tom Cech, and not the graduate students, the postdocs ended up getting a Nobel Prize for this. So, and then that was the second time I visited his lab. The whole outside, and this was, okay, tell you about how long ago it was, there were a bunch of faxes that had been sent congratulating him on getting his Nobel Prize. And basically said it couldn't have happened to a nicer guy. And he was also, this wet behind the ears prospective graduate student. He was more than happy to talk to me about the stuff that was going on in his lab. So it was really nice. So let's slide aside there. But what they had found was that there were certain RNAs that by themselves could basically chop themselves out of a pre-messenger RNA. The ones that they found were actually these right here, what we call the group 1 self-splicing introns, which are a little different than that spliceosomal complex that we talked about. You actually have to put in an extra guanosine residue, but it was all the RNA which could do this, again, in the presence of this extra GTP. relatively soon thereafter, there was a different group, actually, that found these now so-called group 2 self-splicing introns. These now function in an extremely similar way to what we now know about the spliceosome. Forms a 2', 5' phosphate ester bond, this lariat structure, etc. And if you go and look now at the sequences of all of these U4, U5, U6 RNAs, some of them are really quite similar to the sequences you see in these self-splicing introns. So it's not surprising that the active parts of the spliceosome we know now are similar to some of these self-splicing introns. Why bother having all these separate ones? Well, it turns out that you're not going to have a very constrained sequence in this intron if you're going to do splicing with it. And that's going to really constrain what kinds of splicing you can do. If you can now take all of that important RNA, put it somewhere else in your genome, and have it be able to work on different introns, that allows the cell to have much more flexibility in terms of how splicing takes place. And it wasn't just the fact that it was self-splicing introns, it was also that RNA had enzymatic activity, which was the reason that Tomczyk got his Nobel Prize for this. So any more questions on introns? Sorry, before I move along. Yeah?
Are the function of self-splicing introns kind of related to how transposable elements are like jumping around the genome as well?
So the question is there any relationship, again, I'm paraphrasing here, and correct me if I'm missing it. Is this mechanism, the self-splicing introns, is it related to transposons in any way? The answer is it doesn't seem to be. This is all happening at the RNA level, so it's after transcription has taken place. And the transposons are all moving around at the DNA level. Now, yes, they use RNA intermediates, but it's a DNA change which has happened. So these self-splicing introns are not going to be changing the genome per se. They're only changing the transcript that is coming out of it.
So there's no similarity in like the catalysis of like the DNA actually removing its own sequence to the RNA doing the same?
So again, the question here, again, correct me if I'm wrong here, is there any similarity in the actual chemistry or maybe probably the mechanisms of how this cutting and religating is going on? Turns out, so we talked about transposases before. So these transposases and the site-specific recombinases, those have very well conserved tyrosine residues, and those tyrosines are serving as the OH, which is cutting the backbone, actually rather similar to some of the topoisomerases. But that's very different than using the OHs which you're getting off of your RNA. So the mechanism does seem to be rather different.
So what you're saying is the reason, if they could just self-splice, why bother having all the proteins, because that gives it more specificity in how and where it splices?
So I'm not sure I completely understand, but I guess maybe try and re-explain what I talked about before. So the idea, if you have a self-splicing intron, those sequences have to be very well conserved in the intron so that you can still splice that piece out. On the other hand, if you have a spliceosome, and these are genes that are expressed somewhere else in your genome that act on any intron as opposed to that one site-specific intron, you can have a lot more sequence variability in those, and you can splice a lot more different introns. So it gives you more flexibility. Does that answer your question? Okay. Yes?
Is there any significance to why group 1 is like shaped like a foot?
So why do we have a foot and a hand up there? I think it's because the authors of the textbook were trying to be funny. But the important thing with this is it turns out that those sequences actually are very different relative to each other. So there's not just one kind of self-splicing intron. There are multiple kinds of self-splicing introns. There's nothing that looks like a foot or a hand, no. Unlike the case which we have with the DNA polymerases, which really do have that right hand-like structure. Okay, yes, so this was, the main thing here is that a foot is not like a hand. Now, anyone in anatomy will tell me, yes, actually, they're really closely related to each other, blah, blah, blah. But as far as molecular biologists are concerned, a foot is not like a hand. Okay, so let's talk about what happens at the 3' end of your messenger RNA. So as hopefully we all know, poly A tails. When you've actually got your final messenger RNA, there's this long stretch of adenines which are at the end of every one with a few exceptions, of these messenger RNAs that are going to end up being translated. So how do they get there? How do you get all of these A's added? Well, what happens is you have a sequence in your messenger RNA, and I don't expect you to remember this sequence, but it's a specific sequence in the messenger RNA, and then another sequence a little bit further down. And again, I'm not telling you the sequences because I'm not expecting you to remember them. And then a number of other sequences that are not going to be finally included in your messenger RNA. This messenger RNA, as it's made, is going to have an endonuclease activity, which will cut your RNA, and then poly A addition happens, and you end up with this long poly A tail. So let's look at how that happens. Here we have our RNA polymerase happily transcribing along the DNA, making an RNA. We've already talked about this potentially ad nauseum now. The C-terminal domain, which is phosphorylated, carries all of these proteins that are important for modifying the messenger RNA with it. So they're right next to the RNA as it's being made by the RNA polymerase. So the two important proteins, there are more here as well, are going to be CPSF and CSTF. Again, more alphabet soup. Sorry about that. The cleavage and polyadenylation specificity factor, this binds to that sequence, which is going to be 5' to where you actually have your cut. Then the cleavage stimulation factor will associate with CPSF and the RNA to help cleavage take place. This is not the endonuclease itself. There's actually a whole complex of endonucleases that are there that we're not going to get into. So it stimulates the cleavage that takes place due to the specificity that you get from CPSF. Once you have cleavage that takes place, now you have a 3'OH, and now there's the poly A polymerase. Also abbreviated as PAP, but I'll probably try and always use poly A polymerase here. Poly A polymerase is a RNA polymerase, but it doesn't need a template. It just needs that primer, that OH, and all the proteins that are bound to the end of your messenger RNA. And what it will do is add literally hundreds of adenines. Those hundreds of adenines will then bind to the incredibly creatively named poly A binding protein. And the poly A binding protein associates basically all the way along to the end of this poly A tail. How long this poly A tail seems to vary from organism to organism, and even from messenger RNA to messenger RNA. Nobody really quite understands what length that is before they actually end up dissociating. But it seems to be having to do with the number of these poly A binding proteins binding to the poly A tail. So that's what happens to your messenger RNA. Again, we get cleavage and poly A tail formation. What about termination? How does the RNA polymerase stop? Hopefully, Dr. Bartlett talked a little bit about what happens in bacteria. And there you have a sequence in the RNA which can form this secondary structure, i.e. base pairs can form this stem loop structure, and then a stretch of uracil residues. And the formation of this structure in the RNA seems to pop the RNA out of the RNA polymerase. And as soon as the RNA comes out of the RNA polymerase, that RNA polymerase falls apart, can eventually bind to a sigma factor and go and find a new promoter. Eukaryotic transcription is a little different. This termination seems to be dependent on that formation of a poly A tail. So you form the poly A tail on the messenger RNA, but it was an endonuclease stimulated by cleavage stimulation factor. So now we have a 5' phosphate. What is it missing relative to what the messenger RNA looks like? What did we do at the 5' end? Yeah, I know it was last lecture, which was like forever ago. Cap. So does this have a cap? No, it does not have a cap. If it doesn't have a cap, what is going to happen to it? It's going to get chewed up. All of the really hungry exonucleases floating around in the cell are going to start to chew up this end, which is not protected again because it doesn't have a cap. So chews this up, and it seems to be this chewing process actually goes faster than the RNA polymerase is transcribing, and eventually just chews up the RNA, basically right until you get to the RNA polymerase, and then the whole thing falls apart. So a very different way that you're getting transcription termination in eukaryotic cells as you are in bacteria. Next immediate question, what about archaea? We have no clue. So that's what's happening in transcriptional termination. So to risk sort of, oh, sorry, Olivia.
So the addition of the poly A tail includes the separation of the final RNA from the RNA polymerase, right?
So I would say that again, correct me if I'm not answering your question here. I would say that it's the process of forming the poly A tail which leads to termination. Does that make sense, or still not particularly?
I'm just saying, like, when does the actual final process RNA get cleaved off of?
So the processed RNA has been cleaved, and it's now away from the RNA polymerase. That's floating away in the nucleus. The poly A polymerase is making that poly A tail, a poly A binding protein is binding to it. But that's separate now from the RNA polymerase, which is still transcribing. You know, the RNA polymerase has no idea, again, sorry to over-anthropomorphize here, has no idea what's going on with this RNA which is just made.
So my point is, during the formation of the poly A tail,
Yeah.
it gets cut off from the
Yeah, so your pre-messenger RNA, once you have that endonuclease that is cutting it off, that RNA is now separate from the RNA polymerase. Yeah?
So a clarification. It says here that the poly A signal sequence. Is that what creates the poly A tail, or is there a separate post-translational, translational transcriptional modification where it comes in?
So that signaling site, that's where you have the cleavage and polyadenylation specificity factor. So that's that sequence in the RNA which is going to get that first protein, and in fact it's the protein that moves over from the CTD, which is the connection that we have here. That moves over and is going to now bind to the RNA and bring in all the rest of the factors that will give you that cleavage to take place there. So it's all just going on with that. It's RNA and the proteins that are involved there. So there's no extra changes which are happening there. Okay, so I guess kind of a good lead-in, thank you, to this process where you have the RNA polymerase making this RNA. As it's being made, that RNA, in red here, is going to get capped by all the capping enzymes associated with the tail, the spliceosome, a lot of the proteins here are associated with its tail, and the cleavage and polyadenylation factors are associated with the tail. So all of these are right here, and even that exonuclease, which chews in at the end of the 5' end, which was made available by the cleavage that takes place, that's also associated with the tail. So some people like to talk about the CTD as the tool belt of the RNA polymerase or mRNA factory. It's got all these extra enzymes that are important for modifying the RNA as it's being made. Okay, so this, again, we've got these great pictures. Here's the RNA polymerase cruising along, the DNA making RNA, all the proteins are involved, and along with the CTD. Well, if you actually look where these proteins are inside a nucleus, you would sort of expect, oh, there's all these different RNA polymerases cruising along the DNA. They're going to be spread out all throughout the nucleus. That's not the case. Turns out that almost all of, we'll see later, replication. Actually, no, we talked about replication already. But transcription, all of these modifications, they're all actually happening at individual places in the nucleus. So not everything is floating around willy-nilly. They're in these, again, people will talk about them as factories, where you have conglomerations of all of these different proteins. And so that's supposed to be shown over here on the right-hand side. If you look at, for instance, some of the tailing proteins and the RNA polymerases, and just have a specific way of getting them to fluoresce, which is what you have here, you'll notice there are lots of little spots present in the nucleus. Back here. These individual spots correspond to, we have many, many RNA polymerases and many, many of these modification enzymes. And it kind of makes sense, because if your modification enzymes are associated with it. Remember that RNA, which has been made, the tailed, all that's falling off. You're going to need those same enzymes for the next round of making more of that messenger RNA. So these proteins all seem to get concentrated in particular parts. Oh, sorry, this is getting ahead of myself here. Particular parts of the nucleus. And how does that happen? A lot of these proteins are not just associated with the tail of the polymerase, but also these so-called scaffolding proteins. And all that a scaffolding protein is it holds a bunch of proteins together, probably again, so they don't diffuse away and can't be reused for the next round of RNA modification, transcription, et cetera. And we now know that that's what's happening in many of these cases. So whenever we have these pretty pictures, again, in textbooks and my pictures here, we're going to be showing, oh, we've got this single RNA polymerase and this single factor, et cetera. They're all really pretty glommed together through these scaffolding proteins and then parts of the nucleus that are important for this process. Yeah?
So back, how many lectures it was, when you showed the picture of the DNA that has the specific chromosomes in specific areas of the nucleus, is there any correlation between the type of protein that's coded for in those genes and where the RNA is in the nucleus?
Oh, so again, I'll try and get your question here. But the question is if the genes that are coded for on particular pieces of DNA, are those going to be localized at particular positions in the nucleus? What seems to be is actually kind of the opposite to that. It's not where those genes are. It's the genes that are being transcribed that get moved to specific places in the nucleus.
They don't just stay where they are.
Yeah. There actually seems to be movement to these specific places in the nucleus where you have transcription and RNA modification going on. Or if we're thinking about DNA polymerization, where replication is happening, those things get moved to that particular position, which is really kind of amazing.
It seems really counterproductive.
It seems a little bit counterproductive, except if you think about the fact that these proteins are coming on and off all the time, and if you have them all in one place relative to each other, they've got less far to go to find the next gene which is going to be being transcribed, or the next part of DNA which is going to be replicated, the next origin of replication, for instance. So it seems to be that it's more efficient to keep the proteins in one place and move the DNA around. At least that's what we see. Okay, so that's what's going on here. Now we've capped our messenger RNA, we've spliced our messenger RNA, we've put a poly A tail on it. This is great and wonderful, but all the translation is happening out in the cytoplasm. So now we need to get that processed RNA outside the nucleus and into the cytoplasm. How do we do that? Well, one of the things is that cap binding complex that we mentioned way back when we were talking about capping on Monday. That's one part of the process that you have to have. So here's our cap binding complex here in our messenger RNA. I didn't talk much about these exon junction complexes. Exon junction complex is a set of proteins that bind to spliced junctions. So if you have a splicing interaction that takes place, again, you have the whole spliceosome, make the lariat, remove the lariat, the exons get put together. Exon junction complexes will bind to that and basically serve as a little marker to say, yes, splicing took place here. So exon junction complexes have to be there. We already talked about SR proteins because those are the ones which are important for using some of those exons. And then we have HNRNP proteins. Okay, what the heck with HNRNP? So heterogeneous nuclear ribonuclear protein complexes. Most of those are bound to introns. If you have an RNA that has a bunch of HNRNPs bound to it, what does that mean about that RNA? It hasn't been spliced. If it hasn't been spliced, do you want to be getting it out of the nucleus to translate it? No. So another quality control is saying, hey, have we completed our splicing? Two ways to figure that out. One is the presence of EJC, and then the lack of a lot of these heterogeneous nuclear RNPs. It's not all of them. It's just most of those heterogeneous nuclear RNPs. So now you have cap binding complex, SR proteins, EJC. Most of the HNRNP proteins are gone. Now transport takes place. And this is an active process through the nuclear pore where there are RNA export proteins, supposed to be shown here as this excuse me, little mushroom structure, that will bind to properly processed, again, capped, spliced, tailed RNA, and then allow this to be exported into the cytoplasm, at which point this starts to be translated directly. We'll talk about translation, but one of the main things as far as translation is the removal of this cap binding complex and the incorporation of the translation initiation proteins. Then these proteins, the cap binding complex and this nuclear exporter, once it's not bound to the RNA, goes back into the nucleus, finds another messenger RNA, and those get exported back out. We can see this in some cases in the electron microscope. This is a cartoon of it. Here we have an RNA that's being made, processed, all these proteins that are associated with it. That then gets transported out through the nuclear pore complex, and these are things you can literally see in the microscope. That nuclear pore complex is, and hopefully, how many of you are taking cell biology again? A few of you? Yeah, so hopefully you've talked about the nuclear pore complex and nuclear export. We're not going to talk more about it here. More interested in it, take cell biology next term. So a couple of people have asked me in office hours, what happens to that RNA in the introns? And remember, introns are usually way bigger than the exons. So you've got a lot of extra RNA floating around inside the cell. The process that takes care of this is something, again, I think a horrible name, but it's what everyone uses, is the exosome. What the heck is an exosome? Well, it's mostly a whole bunch of exonucleases. 5' to 3' exonucleases, 3' to 5' exonucleases. And this is what I sort of, I kind of jokingly talk about the hungry exonucleases inside the cell. This is really what that is. And so this particular complex will chew up any extra RNA which is left around inside the cell. That then RNA can be reused for transcription, et cetera, later on in the process. And we now also know the high-resolution structure of this. Multiple different, again, 5' to 3' and 3' to 5' exonucleases, which are going to clear up our extra RNA. So, have to have another clicker question, don't we? Actually, I need a drink. That's the main reason to have a clicker question. Messenger RNA needs to have which of the following in order to be exported? A cap, alternative splicing, introns, all possible exons, or HNRNPs bound to it. Yeah, and talk to your neighbors. Ten. 5. It's amazing how those numbers go up as soon as I say 10. It's just the last click that's the one that counts. So it's really, if anything, with the clicker questions, if you've got a gut feeling on it, click it, and then you can go back and change it later. So just some strategies in terms of doing clicker questions. Still don't have 100%. We're still working on that 100%. Yes, it has to have a cap. The key to this question is needs to have, or is required. So alternative splicing happens, but there's no way to control for alternative splicing. You don't want to have introns, so that's out. All possible exons, if you have alternative splicing, you're not going to have all of the possible exons. And then some HNRNPs are bound and some HNRNPs are not bound. So even if you didn't know cap, you could eliminate all of the other ones. Okay, I got rid of it. So finish up talking about transcription, at least in terms of the process of transcription and RNA processing. We'll talk about regulation later on. I'm sure all of you remember the most prevalent RNA inside the cell is what? Ribosomal RNA. And that's because, as we'll see when we talk about translation, the ribosome is basically RNA. Few proteins, but the vast majority of it is RNA. And that has to be active just by itself. So ribosomal RNA, as again, hopefully Dr. Bartlett talked about, in eukaryotic cells is made by RNA polymerase I, and that doesn't have a CTD, but basically the binding transcription factors, general transcription factors, et cetera, they're specific to RNA polymerase I, and will get it to bind to the promoters and give transcription of this RNA. Well, how do you get 80% of your RNA to be ribosomal RNA? There are a couple of different ways of doing it. In bacteria, you have really, really, really good promoters. So the RNA polymerase binds there and will transcribe them all the time. In a number of bacteria, particularly E. coli, you've got about 7 of these promoters. So making lots of ribosomal RNA. Human cells, in fact, most eukaryotic cells, the way they figure out how to get a whole bunch of RNA is have a whole bunch of promoters, lots and lots of copies of these ribosomal RNA genes. So in humans, it's about 200 of these. What does that gene look like, or particularly what does the primary transcript look like? It's this big, almost 13,000 nucleotide long transcript. It's not capped, you'll notice here. Why is it not capped? Because there's no CTD. You don't have all the capping enzymes coming along with the polymerase. So it's a non-capped RNA, and then instead of introns here, the blue bits are the bits where actually are maintained. But instead of fusing all of these together, like you would have with the splicing process, and again, all the splicing enzymes are hanging out on the CTD, so you're not going to have splicing here. These all get chopped apart from each other. And one of those choppings, and I actually don't have it listed on here, is that mysterious U3 siRNA. You remember we need U1 and U2 and 4, 5, and 6 for splicing? Where the heck is U3? U3 is actually here involved in some of these cleavage events, in chopping up these precursor ribosomal RNAs into little pieces. The other thing which happens to these ribosomal RNAs, which is really very different, is this RNA is covalently modified, and in many, many, many ways. This is just a slight description of some of the literally hundreds of modifications that happen to the RNA before it actually can be a functional ribosomal RNA. So lots of modifications. We'll see how that modification happens here in just a second. And then these 3 ribosomal RNAs, which come from this precursor RNA, and then there's another small RNA, which is made by RNA polymerase III. All 4 of these RNAs come together to make the final ribosome. So we'll look at this modification here in just a second. It's these SNO RNAs that are important for that. But before, I wanted to show you this really amazing micrograph as far as I'm concerned. This is one of the regions in, if I remember correctly, this is a frog genome, which is being actively transcribed in the ribosomal RNA region. So each of these little Christmas trees here, so the really thin gray line that's probably a little hard to see here, that's the DNA in the region of the genome where there are multiple copies of this ribosomal RNA gene. And then each of those RNA genes is right here. Transcription starts at this end. It's finishing at that end. And each of these pieces, the branches of the Christmas tree, are these precursor ribosomal RNAs which are being made and then modified, et cetera. So how do we get these modifications? This is the SNO RNAs. SNO RNAs, not to be confused with the SN RNAs. Why SNO? That's the small nucleolar RNAs. And the nucleolus is where you have all the modification that is taking place to make your ribosome. So the nucleolus is a subcompartment of the nucleus that you don't actually need all these, super duper stains to see. You can literally see a compacted part of the nucleus where all of the ribosomes are being made. All the ribosomes are being put together. But what's in there and what's doing these modifications? These are the small nucleolar RNAs, and not surprisingly, they're not just RNAs, they're RNPs. They've got proteins that are associated with them as well. What do they do, and why do you have an RNA as part of this process? What's so important about RNAs? Why do we need RNAs in spliceosomes? What's he putting his arms together for? Base pairing interactions, specificity. How do you interact with another RNA? You interact with another RNA via base pairing. So the ribosomal RNAs, where you're going to get these modifications, and the modifications have to be at very specific positions in the ribosomal RNA. How do you make sure those modifications happen at the right place? You have an RNA which gives you the specificity. So base pairing interactions. That's what's shown down here. We've got our SNO RNAs here together with the protein. So the SNO RNA is in blue. The protein is here in, I guess that's what, reddish, pinkish, something like that. Different SNO RNA with different base pairing interactions. So the SNO RNA tells the rest of the machinery, this is where you need to make a modification. The modifications are made by the proteins, which are there. The most common modifications are pseudouridylation. Pseudouridylation, what the heck is that? So all that pseudouridine is a uridine that's just been twisted one part of the ring. So here we have, normally your uridine is going to be attaching to this NH. That'll be where your glycosidic bond is. Now it's been rotated. It's actually not, that's not the way it's formed, but relative to the structure, this is now where it's bound to. But now we've got, of course, different base pairing interactions that can happen. Normally base pairing can happen here. NHs can undergo base pairing as well. So pseudouridine, one of the most common modifications. The second one is the methylation of the 2' position in the ribose. These two literally are happening hundreds of times through your ribosomal RNA sequences. And again, at very specific places, that specificity, again, comes from these SNO RNAs. Two major families of these are ridiculously named. The box CD SNO RNAs, and that's because they have these RNA sequences, not the ones which are binding to the ribosomal RNA, but those that bind to the protein, which are very well conserved. Those are the CD boxes. And then this one's just a little bit easier to remember, the hairpin ACA SNO RNAs, hairpin because it forms a hairpin structure. And give you one guess what the sequence is right next to that hairpin. ACA, great. So, and this one's important for pseudouridine incorporation. So again, the important thing is we've got all these modifications that are happening in the ribosomal RNAs. Where those take place is due to the SNO RNAs. What does them are the proteins that are part of the SNO RNPs, the P part of those RNAs. Ready to answer a clicker question? Is that what I have on the next slide? No. I've got one more before I do that. Didn't remember where I put them all. So we'll have one right after that. And then, everyone can run away. So if we look at the nucleolus, this is, again, it's a separate part. It's supposed to be this light blue thing here in the middle. It's usually nowhere near this big relative to the nucleus. But you have the DNA and all of these RNA coding genes. So this is that exception to what you were asking about before, whether you have the genes in the place where they're making what's going on. Now we do have all of the genes. And it turns out they're on different chromosomes. So you've got different chromosomes that have these individual pieces right in the nucleolus. So those are present in the nucleolus. You have transcription that takes place. All the SNO RNAs will modify. We have cleavage. And then one of the really amazing things about ribosomal biogenesis is that all these ribosomal proteins actually come into the nucleus, assemble with the ribosomes, and then those ribosomes get re-exported back into the cytoplasm. Really amazing. These are big complexes. And how they get moved back and forth between the nucleus and the cytoplasm is really pretty spectacular here. Turns out that some of the small nuclear RNAs are also made here. So U6, again, the catalytic subunit for splicing, and tRNAs are also being made in this nucleus structure. Even the telomerase RNA is being made here in this nucleolus structure. So, but probably because it's where all your RNA modification is going on. RNA protein association. You've got the proteins. They're there. So you can use all of them in this place. So now we'll have our clicker question. Sorry for going a little too ahead of myself here. Which of the following determines the site specificity for pseudouridylation of a ribosomal RNA? U3, snRNA, HACA, SNO RNA, CD box SNO RNAs. 5S RNA, or the exosome? 100%. Come on, let's get 100%. Vote early, vote often. Wow, not a huge jump in scores. You're listening to me. Hallelujah. Did we get 100%? Not quite. We're getting close. We're getting very close here. So that's it for our our mRNA and other processing. Wanted to briefly start on translation in the last 10 minutes that we have, actually 8.5 minutes according to my clock. So translation, this is, again, what happens. Once you've made your messenger RNA, once you've gotten it out of the nucleus, now it needs to be translated. So what are we going to talk about? Today we'll talk about the genetic code and a little bit about open reading frames, and all the rest of this stuff we'll cover on Friday. So what's the genetic code? This was a huge question, and again, it was starting to be addressed right after Watson and Crick were coming up with their structure for B-form DNA. And yes, I will bring back my model a little bit later on here. And the big question was, and as we mentioned before, how do you go from 4 nucleotides to 20 amino acids? And people knew that 20 amino acids, that's what you found in proteins. 4 nucleotides, what you found in DNA. That was why DNA couldn't be the genetic material, because you didn't have enough information in it. So how do you do this? And a lot of this was solved by physicists, mathematicians, who were out of work because they'd already done the Manhattan Project, trying to figure out what was going on here. So one of the very first things that became very obvious was if you're thinking about having a code going from a 4-letter code to a 20-letter code, you're going to need more than one of those letters. And all that you need to do to look at that is say, OK, if we've just got 4 letters, it's the nucleotide code, just 4 to the 1. If you're just using 1 in the code, you can only make 4 different amino acids. If you've got 2, you can make 16. If you've got 3, you can make 64. But 64 is like way more than 20. This seems kind of bizarre. Why would you have so many? But this is what's happening here, is that n is 3, and 3 nucleotides are going to give you 1 particular amino acid. So 3 nucleotides are going to be a codon. So this was pretty obvious very, very soon. The next question, and this took actually quite a long time for people to work out, is whether the code was an overlapping code or a non-overlapping code. So what do we mean by overlapping versus non-overlapping? If you have a overlapping code, and you have this sequence, this is just your nucleotide sequence, AGC, ATCG, with an overlapping code, you would actually have 5 different 3-letter sequences from this original sequence. If, however, you had a non-overlapping code, you're just going to have 2 and a little bit codons. So this is a way more efficient way to use your genetic information that you're getting from nucleotides relative to protein information. So a lot of people thought that this was what was happening, was an overlapping code. But the problem with an overlapping code is if you have a mutation, and when we talked about DNA repair, we see what a horrible molecule DNA is for genetic information because all these changes can happen. If you have a single base which is changed, in this case, it's this C which goes to an A, the transversion that takes place here. With an overlapping code, you now change 3 of your codons, whereas with a non-overlapping code, you've only changed 1. And this was actually done completely theoretically by a fellow by the name of Sidney Brenner, who published a paper that said, basically, you can't have an overlapping code because all the changes that would happen here are impossible based on the number of actually peptides, and this is literally 2 amino acids right next to each other, which would come out, which is a real, I think, amazing paper thinking about how just the fact that you've got 4 letters and 20 letters is going to allow you to understand what's going on with the whole genetic code. So it's completely theoretical. It was shown that you couldn't have this overlapping code. I think he named the paper on the impossibility of an overlapping genetic code. So, but the other thing, of course, now we know through a lot of experiments that I'll talk about on Friday, that this genetic code is non-overlapping, but is also highly degenerate, which should basically be kind of new because you had these 64 codons and only 20 amino acids. And what do we mean by that? Degeneracy. Degeneracy is that a particular codon, which again is just a stretch of 3 nucleotides, those 3 nucleotides sometimes are going to code for one particular amino acid. And yes, do you want me to remember some of these things for a midterm? This one in particular, yes, I would know. Probably know it already. AUG codes for methionine. You notice that there's only methionine and tryptophan, which only have one codon which codes for it. There are others which get up to be as much as 6. Not important that it's arginine here, serine, leucine, etc. So most have 2, some have 4, some have 6. Only 2 of them just have 1. The other thing, which is important about the genetic code, is that in the genetic code, there are also stop signs. And that's the end of a particular stretch of amino acids in terms of your protein. And that's here, the so-called stop codons. And there are 3 of them. And we'll look at how those are actually being recognized a little bit later on. This is the way they present the genetic code in our textbook. I hate the way this is presented. I much prefer this version, which is another way of looking at the genetic code. Here, vertically, we have the first letter in any given codon. Horizontally, we've got the second letter. And then each of these vertical ones is split up into the 4 different nucleotides. I already mentioned some have 6, some have 1, some have 1. This is the one I would remember. And we have 3 stop codons. But if you look at the genetic code set up in this context, what you will hopefully see is that this 3rd nucleotide really doesn't make a huge difference. Let's just look at leucine. Leucine here, 6 different codons that code for leucine. But any one of these last nucleotides turns out to be one that can be used under these kinds of conditions. The other thing which is important to hear is the color coding. The color coding is that dark brown is going to be your hydrophobic amino acids. The green is going to be your polar amino acids. Dark blue is going to be your positively charged. And then red is going to be your negatively charged. You notice they're all together in the genetic code. And what that means is mutations in a coding sequence, they may even change an amino acid, phenylalanine to leucine, but they're still very similar in terms of their overall kinds of function. So this presence of the genetic code and the grouping of all of these similar amino acid side chains together is probably a mutation protection mechanism. So if you have a mutation that happens, you're going to change. You can even change an amino acid, but it's highly unlikely to change the function of your particular protein. And then, should be obvious here, the first 2 nucleotides of the codon are there. One thing I did want to point out, just really briefly, is that the code is not completely universal. A lot of people talk about the genetic code. This is just one of the genetic codes. And there are some slight modifications to it, some of which have been discovered literally in the last couple of years. So we'll continue to look at this. I basically just all the stuff that I told you in just a second here on Friday. See you then.